what I would like you to do now is relax a little bit, think about your case study, the case study 1, the case study 2. These two are the most interesting part of the whole Gyan session in my viewpoint. What I would like you to do, and I think I have actually had asked this on the very first day, that you do need to divide yourself into groups. So, for the case study 1, case study 2, on the very last day, that would be the Friday, you will be presenting a couple of slides from each and every group. So, if you divide yourself into 4 groups or 5 groups, depending on that, you will be presenting a slide on your case study 1 for the case study 1 session, and on case study 2 for the case study 2 session, and we will have a conversation and interaction. You will be questioned by your colleagues, and you will also be questioned by me. Why you chose a specific host? Why did you choose a specific reactor design? Why did you choose these parameters? What was the intent? If you are choosing 10 degrees Celsius, what is the reasoning for 10 degrees Celsius? You should have answers to those type of questions, and if there is something missing, I will try my best to answer those part of the questions for you. Okay, and that will be a very interactive session. So, my humble request is, do divide yourself into groups, and give your group a fancy name, not something that would be hurtful to others, but something that will add value, like something that you will be interested in. To, like, let's call yourself as a startup in that case. So, give that a startup name, and then we can go ahead from there. Okay? But this part of the session, I'm just going to give you a brief idea of what I'm expecting for your case study 1 and case study 2. Okay? So, the case study 1 was developing a plant protein. So, the protein that I wanted, or I want you to develop, is monolin. Now, monolin is a single alpha helix packed against 5 antiparallel beta strands in a beta grass fold, cysteine fold, is 1500. The most important part of the monolin is 1500 to 3000 times sweeter than sucrose. So, it's an alternative to sugar, not like stevia, better than stevia. It's a protein. Okay, it's a plant protein. I would like you to develop a process. One which you are seeing there is the one which I would be using. So, the host microorganism that I would have chosen would be that's a yeast. I have the amino acid sequence. I have not provided you a DNA sequence. You need to tell me what would be the sequence for your plasmid or whatever process you are using for inserting your DNA into any host, and then how are you going to transfer it into your reactor, what would be the feedstock in the reactor, do you need any growth factors or nutrients, what would be those nutrients. You would be able to identify or find those information online on Google. You don't have to go through a tons of research articles. Even if you do a normal regular Google search, you would be able to identify enough information to answer that question. Okay? And then once you get your product, whether it is intracellular or extracellular, depending upon the host you choose, and what would be the purification and downstream processing steps that you will choose. And to add a little complexity to it, I would like you to perform a simple TEA, which is your techno-economic analysis. In a techno-economic analysis, you look at what would be the cost of your raw material, what would be the cost of the handling of the raw material, whether the technology exists, if it is available, readily available in the market. So let's say if you're looking at sonicator for purification or extraction of your intracellular protein, then bless you. If you're looking at the intracellular protein, you are going to use sonication to do the cell lysis. What is the cost of a sonicator? You identify that on Google. I'm not asking you to give me the perfect value to it, but I'm asking you to give me an idea how you will approach. Because the problem is, what I want you to understand is that when we develop a particular product, it's not just the value of the product depends on the process itself. So if I have a very costly process, but I also have a super costly or super valuable product, I would love to invest because I'm still making money, right? So if monolin is going to replace the whole sugar industry in the food industry, if I'm going to capture the whole food industry, why will I not invest even if the process is costly? I'll do that. So that techno-economic analysis, that's why I want you to work in groups, because then you can divide the different stages of the production between yourself, and then you can identify the economic analysis for those sections. And then you are going to present it to us on the very last day as part of the session, okay? And then we can have a very good conversation why your product costs more and their product costs less, and we can look at, you can have a competition who is going to sell me monolin for cheaper prices, okay? So the benefits of monolin is the digestion has no impact on the blood sugar level. That's why I'm very, very interested in it. But this is the biggest constraint. A heat treatment over 50 degrees Celsius at low pH denatures monolin. Question for you then, how will I use it in food industry? Because the products, let's say if I'm adding sugar to a juice, I'm going to add it before the pasteurization or after the pasteurization? If I have to maintain it, the structure, what if I add before? If I'm adding it as a natural sweetener at home, and then as I'm making tea, I add sugar before. As the tea is boiling, I'm adding the sugar to it, right? So the protein will denature. What will you do to prevent that? Think about it. Okay? So that can be a part of your solution. People have actually answered this during the day session. How will you preserve something which is thermally sensitive? You just take a starch, that's the max hint I can give, and then you do whatever you want, okay? The second case study that we are going to discuss on Friday is developing a microbial protein. Now here, we are going to work with alpha amylase. So you are going to produce alpha amylase, which is an enzyme. So all enzymes are proteins, but not all proteins are enzymes. So I'm taking the benefit of that and giving you an enzyme. How will you get alpha amylase? So there is a process that I would use. If you are going to use the same process, I don't mind. If all the groups are going to use the same process, I won't mind. You can change it if you would like to. But I would love to know the host that you are going to use to grow these for the fermentation process, and what would be the purification process that you will follow for that, okay? Alpha amylase in bacteria used for breakdown of starch as source of energy. It is used in production of food, detergents, bioethanol, and paper. If you remember the example that I gave you, when the cats pee in specific area you have the urine. Amylase is also used for degradation purpose because it acts as a detergent too, right, for cleaning and removal of the aroma. So, in bread for reducing hardness, enhancing elasticity and delaying stalling. And for the gene modification, the gene modification would be required to get certain properties to the enzymes, right. We do directed evolution because we want to change the property of the enzymes. We want to make it much more stable. We want to make it viable at higher temperature ranges or pH ranges, right. So, depending upon what sort of process you will choose, you would be affecting the property of your enzyme. I am choosing alpha amylase because that's a very common enzyme, and most of you, even if you are from the background of food, you will have some understanding of alpha amylase. Okay? So, use that, and I wish you all the luck for that to work in a group and come up with the solutions. So, do you think these two case studies are good enough for you, or you want another one? 2 or more? More than enough? Okay. So, I will keep it at 2. And some of the tools that you can use for yourself would be the BLAST, if in case you are trying to identify the sequence. For monoline, I have only, if you have taken a picture of my slide, you would only get the sequence of the amino acid. You are not getting the sequence of the DNA. You have to find the sequence of the DNA. Okay, so take as many pictures as you want. And if I am there in that picture, I will not mind that. But anyways, so BLAST, FASTA, you can think about brushing up your bioinformatics skills a little bit. If not, you can look into YouTube how to do certain things, or if you can take help, make your group in such a way that somebody knows what to do. Okay. You can use the PDB database bank. All enzymes are proteins, but not other way around. So, if you want to know more about alpha amylase, where will you go? PDB. Okay. If you want to know more about monoline, you will go PDB. So, use these tools and databases to gain more insight. You will also be able to find the source who would have worked first on monoline. You can read those research articles, those are freely available, or you can look into those research articles, how they got it.